Bioremediation. Introduction. Bioremediation is a branch of biotechnology that employs the use of microorganisms in the removal of contaminants, pollutants, and toxins from soil, water, and other environments. Bioremediation is used to clean up contaminated groundwater or environmental problems such as oil spills. Bioremediation is also called as biorestoration, bioreclamation, and biotreatment. Bioremediation relies on stimulating the growth of certain microbes that utilize contaminants like oil, solvents, and pesticides for sources of food and energy. Bioremediation mechanism. Microorganisms destroy organic contaminants by using the chemicals for their growth and reproduction. Waste materials are examined and certain bacteria are isolated based on their efficacy at digesting and converting the waste. Indigenous or local bacteria is to be used. The bacteria then go through several steps of cultures and process for performance testing. The suitable bacteria are placed back in the waste environment. Bacteria will grow and thrive in the process, and then it digests and converts the waste into carbon dioxide and water. The right temperature, nutrients, and food must be present, and conditions are improved by adding amendments. Graphical representation for bioremediation mechanism. Bioremediation is a triple commerce process. Pollutants in soil, water, and air environment are removed with the help of microorganisms and the enzymes produced by microorganisms. Methods of bioremediation. Bioremediation can be done through two methods. That includes in situ bioremediation and ex situ bioremediation. In situ bioremediation. In situ bioremediation, contaminants are treated at the same site using biological systems. This remediation process is less effective and this process is less expensive. In situ bioremediation is mainly used to treat contaminations in soil and groundwater. The remediation rate and the effectiveness of the process depend on the type of contaminant concerned, site-specific characteristics, contaminant distribution and concentration, concentration of other contaminants, microbial community of the site, temperature, pH of the medium, moisture content, and nutrient supply. Examples of in situ bioremediation includes biostimulation, biosparging, bioventing, and bioaugmentation. Biostimulation. In biostimulation, nutrients are added into contaminated soil. It stimulates the growth of microbes, and then the microbes will eat the contaminants. Finally, non-toxic end products are produced. Biosparging. Biosparging is an in situ remediation technology that uses indigenous microorganisms to biodegrade organic constituents in the saturated zone. In biosparging, air and nutrients are injected into the saturated zone to increase the biological activity of indigenous microorganisms. Biosparging can be used to reduce concentration of petroleum constituents that are dissolved in groundwater, adsorbed to soil below the water table and within the capillary fringe. Then, constituents adsorbed to soils in the unsaturated zone can also be treated by biosparging. Bioventing. Bioventing is an in situ remediation technology that uses microorganisms to biodegrade organic constituents adsorbed on soils in the unsaturated zone. Bioventing enhances the activity of indigenous bacteria and stimulates the natural in situ biodegradation of hydrocarbons in soil by inducing air or oxygen flow into the unsaturated zone. Bioaugmentation. Bioaugmentation is the practice of adding cultured microorganisms into the subsurface for the purpose of biodegrading specific soil and groundwater contaminants. Microorganisms are added in contaminated soil. Then the microorganisms will eat the contaminants and finally, contaminated environment is turned into contaminant-free environment. Ex situ bioremediation. In ex situ bioremediation, contaminants are treated in some other place from the original site. That means ex situ bioremediation process is performed out of the location where the contaminants were found. This process is more effective, but this process is expensive. Contaminants are excavated or pumped out from the original site and treated inside the controlled environment. A wide range of hydrocarbons is purified by ex situ bioremediation. Examples for ex situ bioremediation includes land farming, composting, biopiles, and bioreactors. Land farming. Land farming is an ex situ waste treatment process that is performed in the upper soil zone. Contaminated soils, sediments, or sludges are incorporated into the soil surface and periodically turned over or tilted to aerate the mixture. Contaminated media is usually treated in lifts that are up to 18 inches thick. When the desired level of treatment is achieved, the lift is removed and a new lift is constructed. Composting. Compost bioremediation refers to the use of biological system of microorganisms in a mature cured compost to break down contaminants in water or soil. Microorganisms consume contaminants in soil, water, and air. The contaminants are digested, metabolized, and transformed into humus and inert bioproducts such as carbon dioxide, water, and salts. Initially, organic materials used for compost include a mixture of brown organic materials and green organic materials. Brown materials supply carbon while green materials supply nitrogen. The best ratio is 1 part green to 1 part brown material. Oxygen is needed to support the breakdown of plant material by bacteria. Bacteria decompose the material and they release heat, water, and carbon dioxide. Finally, the compost is produced and it can be used as fertilizer for gardens. Biopiles. A biopile is an ex situ bioremediation technology in which excavated soils are mixed with soil amendments, formed into compost piles, and enclosed for treatment. The basic biopile system includes a treatment bed, an aeration system, an irrigation or nutrient system, and a leachate collection system. Moisture, heat, nutrients, oxygen, and pH are controlled to enhance biodegradation. Bioreactors. It is an ex situ process treatment which is done by pumping out contaminated water or soil. It is also known as slurry reactors or aqueous reactors. By engineered container system, it bioremediates the contaminated water or material by reactors. As the physical environment is easily controlled and manageable, the bioremediation rate is more in bioreactors. Bioremediation of marine oil spills. It is a useful process for removing marine oil pollutants. Marine oil spills are mainly oils, petroleum, fuel, etc. Marine oil spills are composed of complex hydrocarbons. Oil spills in the ocean have a severely negative effect on marine life, especially seabirds and filter feeders. Seabirds such as seagulls and ducks spend most of their life on water and go to land only during their nesting period. The feathers of many seabirds are wettable and must be carefully preened for flight. If feathers come in contact with oil, the seabird ingests the oil while trying to preen. If ship moving in the sea hits the glacier rock, oil spills from the ship, then the oil is cleaned up with the help of Pseudomonas and Flavobacterium, etc. Finally, contaminant-free environment is obtained. Bioremediation of groundwater. Pollutants removed by using physical, chemical, and biological or a combination of these three processes, helps to remediate the water, which is said to be groundwater remediation. It can be subdivided into two categories, which include ex situ and in situ technology. Ex situ bioremediation of groundwater. Ex situ technology involves treatment of groundwater by dewatering the polluted water from the aquifer, then treating the water on surface by physical, chemical, or biological technology, and finally re-injecting the treated water to the aquifer. In situ bioremediation of groundwater. In situ technology treatment of groundwater is done within the aquifer by using thermal, chemical, and biological treatment technology. It involves the treatment of groundwater without extracting the water from the aquifer. Some contaminants like dense non-aqueous phase liquids, light non-aqueous phase liquids, inorganic chemicals like ammonia, cyanide, and fluoride, metals, bacteria, and virus are removed with the help of the aquifer. Environmental factors required for bioremediation. Composition of microbial cell. Nutrients are the basic building blocks of life and allow microbes to create the necessary enzymes to break down the contaminants. All microorganisms need nitrogen, phosphorus, and carbon to secrete the essential enzymes to break down the contaminants. Microbial cell is composed of various elements. Carbon is the most basic element of living forms and is needed in greater quantities than other elements. In addition to hydrogen, oxygen and nitrogen constitutes about 95% of the weight of cells. Phosphorus and sulfur contribute with 70% of the remainders. The nutritional requirement of carbon to nitrogen ratio is 10:1 and carbon to phosphorus is 30:1.